So the next session is on electronic health records and the delivery of this information and the use of electronic health records to further stratify risk in various ways. So the first talk is Josh Denny on phenotype risk scores, the other PRS. We had a big debate when Josh was inventing the phenotype risk scores what we should call them. We wanted PRS, but he sent me an email earlier today saying he was happy we decided to use something else because that would have been really confusing.
We thought we'd try to fight the battle of names, but now the challenge is say PheRS instead of PRS. Yes, I will. Is this where? Oh, lean in. Yes. All right. Thanks. So we're talking about phenotype risk scores. All right. So I'm going to start with a case. And so here's an individual that presents at 32 with hearing loss and numbness. Develops called having auditory neuropathy syndrome, blurry vision, muscle spasms, neuropathy. Was confined to a wheelchair, dysphagia, has a peg tube, and respiratory failure, intubated, and is referred to the Undiagnosed Disease Network, where many of you are familiar with the process, which is very intensive in terms of workup and evaluation and careful exams, including sequencing. We'll come back to this. So, where we are, so, the idea of GWAS and PheWAS, and we've been talking a lot about PRSs, and so now we're going to talk about phenotype risk scores and the process here. And so in this case, we're going to aggregate phenotypes just like we aggregate genotypes in polygenic risk scores. So the process for doing this is to go through, and we started with a knowledge-based approach that goes through and uses OMIM and the human phenotype ontology to go from a disease and a gene linkage into a number of phenotypes, and those phenotypes can be aggregated and matched to phenotypes in electronic health record. And we started using the PheWAS approach. So the PheWAS approach is a groupings of ICD-9 and ICD-10 codes into about 1,800 phenotypes. And so by aligning those 1,800 phenotypes with the several thousand HPO terms, we're able to create a map that can be iterated across on really sort of any Mendelian disease for which there are HPO terms defined. And of course, that could extend beyond there as well. And so we also create a weight for each feature, and in this case we use the prevalence essentially in the electronic health record system as a way to weight the phenotype. And then you can just iterate across Mendelian diseases. And so looking at cystic fibrosis, here is a list of features from OMIM, and those can have corresponding human phenotype ontology terms, and those can be mapped to PheWAS codes, which give you, these numbers look like ICD-9 codes, but they're not. They're the fact that we started with ICD-9, and that's why they kind of look like them. But they map to groupings of ICD-9 or ICD-10 code. And so if you take a series of patients that have features of cystic fibrosis, you can go through each of these features. They each have a weight, and you can see, asthma doesn't have as high a weight as bronchiectasis, and bronchitis using the way we originally chose to weight these. And so when you sum these up, you get different weights for each individual. And when you look to try to see whether or not this works, we started with looking for people who are diagnosed with the condition and those who weren't. And so, you notice that we just use the features of disease, not the disease label. So this is just using bronchiectasis, asthma, pancreatitis, things like that. And you can see that it effectively separates his cases from controls based on the score. And so we started with CF, and we picked, when we started this, 5 other conditions. Remember, it's just based on the features, so and not the disease label. So for 5 of these 6 diseases, typically they are manifesting features, but for PKU, it kind of serves as a negative control, because if you avoid phenylalanine exposure, you don't get the features of the disease by and large. And so they don't have elevated scores. We have, of course, found people that have elevated scores for PKU that have symptoms, but by and large, when we did this and found cases and controls, the patients were doing well and avoiding environmental exposure. And so then we turned it on and looked at a number of genotype that we had. So we looked at data from our exome array. These were about 6,000 variants that occurred at 1% or less that really, aren't necessarily known to have any effects. A very few number are P or LP variants, but most are going to be variants that were just picked and put on an array. And in this, we found 18 significant associations, 14 of which were novel associations. The colors there represent the different phenotype domains. So you can see, most of these diseases, a feature was that most of them had different phenotypic features in multiple domains: respiratory, cardiovascular, neurologic, etc. And all of these had at least 3 features in their score. That was one of our requirements. So it's one of the methods there. And then we went back and sequenced several of these that were novel, and then on the bottom we have cystic fibrosis as a reference point amongst those people who are diagnosed. On the left shows the variants that were that led us to that were significant associations that led us to sequence these individuals. And then if they had a second variant found, we annotated those. And the x-axis here is z-scores from their expected phenotype risk score given their age and sex. They were all European ancestry. What you can see here is, the z-scores for those with cystic fibrosis tended to be, 4 or greater standard deviations from expected. On all those individuals were diagnosed, which is why they're squares. You don't see any other squares, and so no one else was clinically diagnosed. And you can see that many of these individuals have very elevated scores. And some of these had pretty dramatic clinical impacts, such as the 7 to 51 with kidney transplants. We also had another HFE variant. 4 of those 40 individuals had liver transplants. Another thing you can comment on here is for many of these individuals with one variant and considered recessive diseases, there was some risk for heterozygosity for the allele. So we also did PheRS on these, and the bolded phenotypes here are phenotypes that are part of our phenotype risk score. So remember, that's a knowledge-driven approach, not a sort of PheRS-driven approach for what we put in there. And so there's a lot of features here, and so this is looking at one of the CF variants. You can see that you put together a lot of those different features and you have a very high score for cystic fibrosis. But there's a lot of other things that other phenotypes that are highly associated with, like MRSA and pseudomonal pneumonia, things like that are very associated with disease. Here's cystic fibrosis as a disease label, and there's the phenotype risk score. P-value is certainly not a perfect measure of sort of predictive ability, but at least you can see that the phenotype risk score does well compared to the cystic fibrosis diagnosis as far as predictive value. So this was a CPC case that came out right around Christmas time that Dan sent us, and he saw it. He read his New England Journal email faster than I did, and so this is a 47-year-old who had a number of recurrent features of cystic fibrosis. And this is highlights that I did, and many of us are aware of conditions that happen of older individuals that are diagnosed with CF, not just in childhood. We calculated the phenotype risk score for this presentation. It'd be in the top 2 percentile of this individual, and went back and when I say we, I almost always are meaning Lisa Bastrach in this case, who has been doing this work, and reviewed all the charts of those individuals who were diagnosed at Vanderbilt for CF, and looked at what age they were diagnosed at. So these were people who were first diagnosed at Vanderbilt and the age, and you can see we actually have an individual in the last year that was diagnosed at 80 years of age. Obviously, she's done very well with her CF, but she also, for quite a long time, has had a number of features of the disease, and it does still have implications. Her genetics are different from the people at the bottom, as you would not be surprised to hear. But you can see that individuals are being diagnosed at older ages over time, which probably doesn't surprise any of us. Emerge, we're doing a project where we're looking at 25,000 individuals and 109 genes, 58 of which are ACMG genes, and for return. And so, this gives us an opportunity to use this kind of approach in a sequence population that has EHR data to think about the pathogenicity of variants. And so in this case, we started with known pathogenic variants and benign variants. And again, these are z-scores, so number of standard deviations from expected for that given individual. In some cases, there's more than one individual in each bucket. I'm not sure if there are more than one in any particular bucket. But you can see all the pathogenic variants that we found in the first 15,000 individuals had elevated scores, and the benign variants do not. So that's reassuring for our classifications in ClinVar. And then we looked at the VUSs. And so the first thing to note is, of course, there's many more VUSs, and we all know this, than there are LPs or benigns. And so some of these are red. And so this is just kind of that verification of EHR as a tool to read out for these populations, and amongst sort of sequence populations that were not really selected in the most case for particular disease, and certainly not for Marfan's, and suggest that we can look at these and use that as a readout. So if we go back to our case, she was diagnosed with riboflavin deficiency, and she began megadose riboflavin, and begins to walk without a walker. Not all her conditions are reversible, but she is certainly better and doing better, and have changed the trajectory of her disease. And when you go back and look at her phenotype risk score, it was 14 standard deviations above expected, which is a very low probability, quite enriched. So that's one of the ideas, or one of the scenarios that we think about potentially finding people earlier, like we mentioned with the cystic fibrosis case. And so here's a case of an individual with a JAK2 mutation and their phenotype risk score over time, given and their clinical diagnosis with the red line. And so, by the time they're clinically diagnosed, they're in the 99.5th percentile. This actually is not using lab data at this point. This is just using their conditions that they are experiencing. But I just want to highlight that this is still very hard. There's a lot of these graphs and numbers can look really good, but if you have a 200-fold enrichment, that takes your probability to about 1 in 10. If the algorithm's enriching 200-fold, which is pretty dramatic for any enrichment test. So we're working on this, how to do this and how to do this better. We get some really beautiful AUCs and things like that, but when you actually put it in terms of positive predictive value, how you do on those top 10 scoring patients, how you do on the top 100 scoring patients, that's something we're really working on now in the prospective application, or potential prospective application of this. And like I was saying, we're really just focusing on the right now the billing code data. It's been very effective for phenotype risk scores, as many of you know in our experience with the PheWAS and phenotype risk scores. In Emerge, we spent a lot of time harvesting the richness, the longitudinality of the electronic health record. We've built a lot of complicated phenotype algorithms that we validated, and there's a richness there, and certainly longitudinality with this depth of data that we could leverage. I mentioned the JAK2 scenario, and certainly looking at lab values, and you can imagine those sorts of things, certainly with measurements for Marfan's and things like that, may really add to the story. And so this is a slide that is a little bit old, but some of the numbers are updated, we have a lot of research cohorts out there. One of the translations and importance of these research cohorts is with these rare conditions, how we get them to talk together. And I think there's a lot of efforts going on with a lot of groups that's sort of been alluded to with Terry's first presentation of how we can do this. Having that rich phenotypic information and the way we can cross across different domains, I think will be important for that. But as Ewan Bernice pointed out, the amount of clinical sequencing that we're doing and as part of cancer testing, rare disease testing, developmental delay with recent guidance from Blue Cross Blue Shield and their evidence street that they would, that it seems to make sense to test in those conditions. United Healthcare has made movements in this area. Obviously, we all know the proliferation of cancer testing that's happening, that there's a clinical resource that will grow very large and represents opportunities, especially amongst those who can leverage non-PDF formatted versions of genetic results linked to electronic health records. And so I think that represents a real opportunity as we move forward. And I have this last comment about the learning healthcare system, which we talked a lot about, but I feel like the enormity of the need to interpret incredibly rare variation may be one of those pushing points that could help accelerate more common practice with learning healthcare systems potentially. And so Lisa Basarach is up there on the top, who's done a lot of this work, and a number of others have contributed across the system to make this happen. Thanks.
I think we'll take questions now or defer them for the discussion. Where are we with time? We have 1 minute and 44 seconds.
Yeah, I was just watching that one. That's all I know.
Iftikhar?
Josh, does your algorithm take into account the frequency of some of these ICD codes appearing, or the temporal gap between those codes? Can you weight those and perhaps become even better at identifying?
We do weight them. We've actually done it weighted and not weighted, and we've tried some different degrees of sophistication. I think this is of weighting approaches. What we do is a pretty simple one that's sort of just like we do for search retrieval, term frequency, inverse document frequency approach. And then we do not look at the temporality, though, at this point. There might be some benefits to looking at that. The one way in which we do generate or adjust for temporality is basically the amount of data they have. So we look at number of years that they have data within the EHR. That's been very helpful to adjust for people that just have really long histories and have more incidental exposure to other codes popping up.
Josh, that was great. Using the EMR as a diagnostic tool, it is great, but it's an expensive diagnostic test. And it's useful for finding patients that are well into the lifespan and who have been underdiagnosed. I think we want to get to the point where we make a diagnosis early in life, if not at birth. And so what are your thoughts about that? How can you, going forward, can you somehow take what you've learned from this and move the detection needle much earlier?
I think that's right. I mean, I think the goal would, the phenotype risk score is telling you the impact of the disease on individuals. So you actually want to diagnose them as early as possible when their phenotype risk score is low as possible, right? So you bring up a great observation. And for instance, fortunately, we find that it's not much help at Vanderbilt in hemochromatosis. We started there because as a clinician and internist, I thought, that's one of the ones I thought of. And the good news is everyone thinks of it. And so that's what you hope to find for many of these. So I think it's a great, there's so much we don't know, and as people get sequencing results, for instance, and if we can do more of that, you could go through, it becomes a closed loop, and you learn more about what you don't know over time by people's phenotype risk scores against given diseases and their sequencing results, for instance. The other thing I think about is, maybe you could do something with family history. If we really knew family history, maybe you could leverage things like that to think through this amongst patients, again, that were undiagnosed. And Nick's work is going to probably talk to us about that, I presume.
I think we're going to go on to the next talk. So Rob, hold your thought and hold that family history thought too. So I think that's going to be fodder for more discussion. Mark, Craven is the next talk. Some lessons learned from using machine learning methods to predict adverse health outcomes.